Hi, welcome to Chapter 3. This is Dr. Randy Papke, your biology teacher. Chapter 3 is all about cells, and we're going to learn about several different types of cells. We're going to learn about how things move across cells, in and out, and and basically learn about all of the big parts of a eukaryotic cell. So to start out, we are going to learn about cell theory. Remember, a theory is something that explains a general phenomenon. It's got lots of different explanations built in, lots of different hypotheses built in, and this particular theory, cell theory, has been tested for a very long time. So we have lots of support for it. Here's the thing about this cell theory that number 1 and number 2 right here. These aren't the testable parts of the cell theory. These are the things called tenets, which are the overriding principles of the theory. So there's two major tenets of cell theory, and that's all living organisms are made up of one or more cells, and all cells arise from other pre-existing cells. Now, the second one was really important back in the day because before cell theory came along, there was this idea of spontaneous generation, and they thought that life could just spring up from things that were not alive. And while it's possible for that to happen, if it does happen that way, that's not under the umbrella of cell theory. And if it does happen, you wouldn't get very complicated life out of that. So you wouldn't be able to get like a whole human being from spontaneous generation. So here's what we know about cells. They are the smallest unit of life, and so that means that cells are alive, and that cells make up of all living things. Cells can function independently. That means that, if you take a blood cell from your body, it performs the functions of life. And in fact, all cells perform all the necessary functions of life. It doesn't mean that all cells do all jobs. Here you can see that, blood cells might have one job, and neurons have another job, and muscle cells have a third job, but they all perform the functions of life. And so it might be a good idea to talk about what the functions of life are right now. We'll hit this again when we get into, I think it's chapter 12. But so life involves being able to metabolize. It involves being able to reproduce itself. It involves being able to get rid of wastes and evolve, and there's a bunch of other things there. That's what cells can do independently of the body, but also in a multicellular organism such as yourself. It does those in concert with all the other cells in your body. So I did mention earlier that there are a couple of basic categories of cells, and these are called the prokaryotic cell and the eukaryotic cell. You can see this word here, karyo. That's the root word right there. That means basically cell. So it means basically nucleus. So prokaryotic means before the nucleus, and literally prokaryotic cells don't have a nucleus, but they do have DNA, and you can kind of see in this little diagram here that there's the DNA. It's free floating, meaning that it doesn't have the DNA in a nucleus, but that's okay. You don't necessarily need a nucleus to get along. You definitely need DNA. So, and then there's this eukaryotic cell, and eu means true, which is a nice rhyming word, so eu means true nucleus. And so here we have DNA found inside of the nucleus. And anything that's made up of, any living thing that's made up of prokaryotic cells is called a prokaryote, and anything that's made up of a eukaryotic cell is called a eukaryote. So those are the two basic cell types. We're going to dive into the parts of the prokaryotic cell first and learn a little bit about that before we move on to spending most of our time on the eukaryotic cell. So here's our prokaryotic cell, and you can see that there's definitely a lot of structures here, but it's not too much to learn about. So a fairly simple structure. We're going to start out with looking at the structures that are found in all prokaryotic cells, and that includes the plasma membrane. Now, the plasma membrane is sometimes called the cell membrane, and so you'll hear those words used interchangeably. The thing about the plasma membrane is, and all cells have plasma membranes, including all prokaryotes, the thing about the plasma membrane is that it is like the barrier between inside and outside. And you might think about that as kind of like a wall, but cell walls are very, very different. Plasma membranes are permeable. They allow things in and out, but they don't allow everything in and out. So kind of like gatekeepers. Not cell wall, it's called the plasma membrane. We'll talk about the cell wall when we get over to this side on additional structures. But we're still over here with the 4 structures found in all prokaryotes. The second of the 4, excuse me, is the cytoplasm. There's the cytoplasm word, and the cytoplasm is all this kind of gel-like fluid that's kind of just floating free, no dots, no strings, nothing, just kind of that jelly-like fluid. It's very thick, and that's where all the chemical reactions are happening inside of the cell. The next structure are the ribosomes. Ribosomes are extremely important. They are these little dots in the diagram. So there's a ribosome there, and there's a ribosome there, and there. And what ribosomes do is they take information that's stored in the DNA, and they make proteins. So it turns out that proteins are probably the most busy chemical in your body. They're really doing a ton of different jobs, and so proteins are extremely important. You got to make proteins, and you make them at the ribosomes. The next structure and the final of the 4 that are found in all prokaryotes are DNA. And there's actually 2 kinds of DNA in prokaryotes, 2 basic structures of DNA. They're very, very similar. It's just one's bigger and one's smaller. So this is the chromosome, the big one. And there's really only 1 chromosome in any prokaryote, 1 chromosome. It contains most of the instructions for that cell's functioning. But there are these smaller bits of DNA, and those are called plasmids. Your book in chapter 3 doesn't use the word plasmid, but it does use it in, I'm sorry, we're in chapter 4. The book has changed numbers. Sorry about that. Your book in chapter 4 doesn't use the word plasmid, but it does bring it up in chapter, I think it's chapter 7. So I'm just introducing it to you right now. I do want you to learn it. There's the term plasmid. And the plasmids are these much, much smaller rings of DNA. And they're not part of the chromosome, but they do contain information. So we call this one the chromosome, and we call these plasmids, but they're both DNA. The next is the structures that are in addition to the primary structures, and they're not necessarily found in all prokaryotes. The cell wall is the one that we're going to talk about next. This thin layer right there, that was the plasma membrane. The cell wall is a lot thicker, and the cell wall does actually protect the cell in some ways. It's not a gatekeeper though. Like if anything can kind of pass through the cell wall, how it protects the cell is it protects it from crushing, it protects it maybe from being eaten, it protects it from being digested maybe. It also gives the cell shape. So if you didn't have a cell wall and you were a prokaryote, you would definitely be around a cell, but this one has a cell wall, so it has more of a capsule shape. The next structure are these little string-like things projecting, and I want you to pay real close attention to this word. These are pili. And the thing about pili is people get them confused with cilia. We'll hit cilia when we get into the eukaryotic cell. But for now, pili are just these hair-like projections. They don't wiggle like cilia do. They don't wiggle like flagella do. They're more like Velcro. They just kind of stick out like Velcro, and they allow the prokaryotic cell to attach to things. So in fact, sometimes what these pili do is they attach to other bacteria, and when that happens, they'll do this thing where they exchange DNA, but they're really only exchanging plasmids. So this one will send a plasmid through the inside of this pilus and send it to maybe another bacteria, and they might exchange DNA. The last structure is the flagellum. We see 1 flagellum right here. We see 3 flagella right here. And flagella are what help bacteria move. Not all bacteria have flagella, so not all bacteria can move. easily, but this one can. So those are the structures of the prokaryotic cell. There's some really cool things that I want you to know about prokaryotic cells. For example, the first cells to evolve on this planet were in fact prokaryotic cells. And that kind of makes sense because they're fairly straightforward, fairly simple. We will talk about how this probably happened in a later chapter, but remember that how the first cell forms is not under the bailiwick, under the umbrella of cell theory. So we're not going to talk about it too much right now. When they evolved on this planet, it turns out that there was not a lot of oxygen in the atmosphere. There was hardly any at all, if any. We haven't been able to find much evidence that there was free-floating oxygen in the atmosphere. So that means that these organisms were able to survive without oxygen. And there are still bacteria today that can live without oxygen, and you can usually smell them. They smell like sulfur. So sulfur and oxygen are pretty similar on the periodic table, and prokaryotes some of them are able to use sulfur instead of oxygen for their metabolic processes. Prokaryotes were also the first organism to evolve photosynthesis. We tend to think about photosynthesis as happening only in plants, and we're going to find out later in this lecture that it's because the prokaryotes were able to photosynthesize that in fact plants were able to photosynthesize. So we'll talk a little bit about that in a bit. But in the meantime, this ability of the prokaryotes to photosynthesize allowed the Earth's atmosphere to change. One of the major byproducts of photosynthesis is oxygen. And so these photosynthetic bacteria, these photosynthetic prokaryotes, were able to take in carbon dioxide and basically transfer it into being oxygen. And that's why we have so much oxygen in our atmosphere today, and why we have so little, relatively speaking, carbon dioxide. There are two main groups of the prokaryotes. And we call these groups domains. So we have the domain bacteria, and bacteria are everywhere. One of the most fascinating things that we've been learning about bacteria is about the bacteria inside our bodies. And the bodies, when the bacteria are living in your bodies, we call it the microbiome. So we'll talk a little bit more about the microbiome throughout the semester. But you need bacteria in order to stay healthy. You need them. You don't need the bad kind. The bad kind will kill you. But you need the good kind. Bacteria do so much for us. They make vitamins for us. They digest food for us. We can't actually digest plant material unless we have bacteria in our guts. So that's kind of cool. We're dependent upon them. There's also, we've been able to find bacteria about miles beneath polar ice caps. We've been able to find them at the bottom of oceans. Domain bacteria are basically every single place there is on the planet you can find bacteria. The second group of prokaryotes are called archaea, and archaea means old. And they are old, but not quite as old as bacteria. Yes, you can find domain archaea everywhere, like in your bodies. But, interestingly, they also are very fond of extreme environments. Like, there's pretty much only a few things living in the Dead Sea. The Dead Sea is called the Dead Sea because there's not much living there, but you can find domain archaea there. You can also find some really weird shrimp there. They live in high salt environments. They can live in high temperature environments. Boiling water, they're the only thing that can live in boiling water. So the good thing about archaea is, unlike bacteria, they cannot cause illness. So even if they do live in boiling water, they can't hurt you if you take them into your body. That's good. They are fascinating things, though. So we're moving on. That's all I really have for you for the prokaryotes. We are actually going to talk about the eukaryotes now. And remember how I broke down just now that prokaryotes were in those two domains, bacteria, and archaea. Well, there's actually really only one kind of eukaryotic organism that's made up of eukaryotic cells, and those are the ones that belong to the domain Eukarya. Most organisms in the Eukarya are multicellular, like the ones that you see over here in the picture. There's fungi, and there's mammals and fish and plants and some really weird little worm. All of those are multicellular, but there are some single-celled eukaryotes. If you are multicellular and you're a eukaryote, you're either a plant, an animal, or a fungus. You could also be a protist, but most of the protists, which is the fourth category under the domain Eukarya, like amoeba, like paramecium, those are single-celled protists. So those are the four, call them kingdoms, underneath the domain Eukarya. A quick compare and contrast, because we've already gotten to know a little bit about our prokaryotes, which you see over here, and we're starting to just kind of delve into the eukaryotes. So compare and contrast here. Prokaryotes don't have the nucleus, eukaryotes do. That's kind of built into their names. You also saw back in that compare and contrast earlier when I first showed you the prokaryotes and eukaryotes that prokaryotes are much, much smaller than eukaryotes. Eukaryotes have a lot more structures in them, so they're about 10 times bigger than a prokaryote. So this one looks to be about the same diameter as like this one, but that's because this one is being magnified a lot more than this one is being magnified. So with the eukaryotes, we have all these internal compartments called organelles. There's your term, organelles. And we don't have internal compartments inside a prokaryotic cell, so they don't have organelles. Yeah, a few exceptions, but we're not going to talk about them. And then finally, and the one that your book doesn't mention, but I really like to mention, is that the DNA of prokaryotes is circular, and the DNA of eukaryotes is linear. It's in a line. It has two ends to it. If you just go back really quickly here, remember that we had those circles of DNA. But if we go back here, you can see that DNA of the eukaryote is linear. They're in lines. So there's a lot of structures that we are going to be learning about, and let's just head down this list, just kind of mention them, listen to them for the first time. I'm not going to go into any details yet, but I will before the end of the lecture is over. Oops, sorry about that. The first is the nucleus, and then comes the plasma membrane. These are structures found in all eukaryotic cells. We're only looking at animal cells and plant cells here. We're not looking at the fungal cells. They're a lot more complicated. Remember the plasma membrane, which is also called the cell membrane. That's found in all living things. So of course it's going to be found in animal cells and plant cells. You can see just how thin that plasma membrane is here. It's that inner layer right there. And remember that the plasma membrane allows things in and out of the cell. So it's kind of like the border of the cell. They have ribosomes, just like our prokaryotic cells. Those little dots right there in the cytoplasm and there in the cytoplasm, those are ribosomes. They make proteins. They have mitochondria, which is nicknamed as the powerhouse of the cell. Both plants and animal cells have mitochondria. Kind of remember that. That's really important. There's something called the endoplasmic reticulum, and there are two types of endoplasmic reticulum. There's the rough endoplasmic reticulum and the smooth endoplasmic reticulum. In this diagram, the rough endoplasmic reticulum, you can see, has these dots on it. Any guesses about what those dots are? Because they're the same dots as the ones found in the cytoplasm, which were ribosomes. So ribosomes are rough endoplasmic reticulum. Smooth endoplasmic reticulum does not have ribosomes. So they're going to be a difference in function. In your book, the diagram for the smooth endoplasmic reticulum looks like coral reef. So that might help you differentiate between the structure down here, which is the Golgi apparatus, found in both plants and animal cells. Also, cytoplasm is present. Cytoskeleton they look like these little wiggly lines or maybe these little straight lines in the cytoplasm. We'll talk about the function of those, but just a heads up, it has to do with maintaining structure and a few other things. And then the Golgi apparatus. And finally, there's these structures here called lysosomes that we'll learn a little bit about too. Now, plants and animals do have some different structures. In plants, they do not have centrioles. Only animal cells have centrioles. For now, centrioles are just, they're important in cell division. We'll talk about them more when we get into the cell division chapter. If we look over here in plant cells, we've got this cell wall. Same function as in the bacteria cell, but all plants have cell walls. And then we have chloroplasts. That's the structure that goes through photosynthesis. And finally, we have this structure called a vacuole. Now, vacuoles are large, empty structures. Sometimes you can find those vacuoles in animal cells, like food vacuoles, but they're just temporary structures. Whereas in plants, the vacuole is a permanent structure. So how did we get all of these structures in the first place? Remember how I talked about the fact that that the first cells on the planet were, oops, sorry about that. Hold on a sec. Hold on. Okay, enough of the slide fumbling. Here we go. So, so eukaryotic organisms have all of these structures. Prokaryotic organisms, the first ones to evolve on the planet, have very few structures. How do you evolve from being a prokaryotic cell into a eukaryotic cell? How do these structures form? And there's two basic ways. The first way is called invagination. And invagination is simply when the plasma membrane folds in of itself, and that's actually something that's fairly common to happen. We'll talk a little bit later about exocytosis and endocytosis. So this folding in is fairly common. What we think happened is that the plasma membrane folded in and maybe surrounded the structures inside the cell, and then you get like a nucleus, right? There's the plasma membrane. And it is true that the membrane that surrounds the nucleus is the same exact type of membrane that exists in the plasma membrane. Other structures that are membrane, we call membrane-bound organelles, those inner compartments, is the rough endoplasmic reticulum, the smooth endoplasmic reticulum, the Golgi apparatus, those kinds of things. So we think that because they're made up of plasma membrane and because prokaryotic cells don't have them, that invagination is a very likely phenomenon that caused those organelles to form in the first place. The second way that this could have happened is through a process called endosymbiosis. And endo has the root of inside, and symbiosis means to live in close contact. So endosymbiosis is the process by which we believe that, a prokaryotic cell was engulfed by a eukaryotic cell, maybe for eating purposes. Here you see this process of, it looks like invagination, but it also will look like endocytosis, we'll see later, bringing this cell into the main eukaryotic cell. And we can tell that it's a eukaryotic cell in this diagram because the DNA is linear, meaning it has two ends here, whereas in the prokaryotic cell it's circular, that DNA is circular. So invagination, but once it's inside, the cell did not digest it. Instead, the cell here, this ancestral prokaryote, is living inside of this ancestral eukaryote. And so over the years, whenever the cell divided, all the interior stuff divided too, and you get this structure. This particular prokaryotic cell was very, very, very good at making ATP, adenosine triphosphate, the energy of the cell. And so over the many, many years, millennia, thousands and thousands of years, it became mitochondria. And there's actually two structures inside cells that we believe went through the endosymbiosis process and became organelles. And those are mitochondria and chloroplasts. We think both of them evolved in this process. You might be asking yourself what evidence we have for this endosymbiosis idea. And there's actually some really good evidence for it. So the first one is the size. Chloroplasts and mitochondria are about the same size as your average prokaryotic cell. So that's interesting. Although there are other structures inside of eukaryotic cells that are about the same size. So it's just an interesting piece of information. We start to get in a little bit more interesting evidence when we get into the DNA, because it turns out that both chloroplasts and mitochondria have circular DNA, just like prokaryotes. This is in fact very different from the linear DNA that you find in the nucleus of a eukaryote. Next is cell division. One of the things we have not yet talked about, we'll talk about in a future chapter, is how prokaryotic cells divide, comparing that to how eukaryotic cells divide, and they divide in a very different process. Prokaryotic cells divide by something called binary fission. Again, details on that later, but it turns out that chloroplasts and mitochondria can make copies of themselves inside the eukaryotic cell, and they do it through the process of binary fission. So that's very strong information, that along with the DNA. A little bit more esoteric now on number 4, ribosomes. Chloroplasts and mitochondria have their own ribosomes. Remember that we saw that picture of the prokaryotic cells earlier with ribosomes inside. Here's the thing. Prokaryotic ribosomes are smaller and eukaryotic ribosomes are bigger. Turns out that the ribosomes found inside chloroplasts and mitochondria are the smaller ones. They're the ones that are made by prokaryotes. And finally, this one of the double phospholipid bilayer. Again, we haven't learned too much about the phospholipid bilayer yet. We will in the next video of this chapter. But for now, the double phospholipid bilayer is the same thing as saying the plasma membrane. So right here, you see this eukaryotic plasma membrane. And here you see the prokaryotic plasma membrane. And when it gets engulfed, it literally has two plasma membranes, the one that was originally the prokaryotic membrane and the one that was originally the eukaryotic membrane. So those are, we call that the double phospholipid bilayer because the phospholipid bilayer is what makes up the membrane. Okay, so heads up on that. We got 5 lines of evidence for the endosymbiosis idea, and some of them are really strong evidence. So we're going to stop now and pick up on this, finishing up this chapter in the next video of this chapter.